Okay, so sometimes things go wrong. Sometimes there are alterations in chromosome number or the structure of chromosomes, so accidents happen. Non-disjunction is when a pair of chromosomes fail to separate. So one gamete has too few and another one has too many. In order to diagnose this, we do a karyotyping, which is an ordered display of an individual's chromosomes. To prepare a karyotype, white blood cells are isolated and stimulated to grow. Then they are arrested at metaphase. That's when the chromosomes are all condensed. And then they photograph them under a microscope. The chromosomes are then arranged into ordered pairs so that any chromosomal abnormalities can be detected. An extra copy of chromosome 21, down here, is what causes Down syndrome. About 1 in every 850 children are born with Down syndrome, and they have a shorter lifespan. This is more common with older mothers. So with older mothers, they usually recommend that the fetuses are tested. Most other autosomal abnormalities result in death. So if you get an abnormality from any of these others, then the fetus is not viable and will die. Sex chromosomes, though, you can have an abnormal number. If you get 2 X's and a Y, Klinefelter syndrome occurs, and the person will be sterile with undeveloped testes and some secondary female characteristics because they have this extra X that they shouldn't have. You can get individuals with 2 Ys, and when this occurs, we get a normal male. I believe that they did a study and they found that XYY males were more likely to be in prison, but I'd have to check on that. XXX females appear normal, slightly taller than average, and XO females end up with Turner syndrome. So it is possible to have abnormal numbers with the sex chromosomes. It is not as possible with the autosomal. New species can arise from errors in cell division. Non-disjunction can produce polyploidy. Poly just means many. So we had diploid, which had 2, and haploid, which is 1, and now we've got polyploid, which is many. These are organisms that have an extra set of chromosomes. So for some reason, a diploid, a 2N gamete, is formed in error, and it produces a 4N tetraploid, that's 4-ploid, zygote. This is more common in plants because once this organism is created, it can self-fertilize because it would need to fertilize itself with another with another 2N gamete. And your book details there's a series of frogs that evidently this happened to. There are 2 sets of frogs. They look identical. One of them is diploid and the other is tetraploid. You can also have alterations in chromosome structure, and these can result in birth defects and cancer. So there are 3 different ones we're going to look at. We can have a deletion. This is where part of the chromosome is just deleted. It's kind of left out. And so the individual is missing that little part of DNA. And it depends on what's missing as to how detrimental that might be. We can have a duplication. So this little copy of DNA is over here twice, And having two copies of a part of DNA can also affect an individual. Third, we have inversion. In inversion, a segment is removed and inserted backwards. So this is purple-green. Over on this side, it's green-purple. And the last one we have is reciprocal translocation. And this is where non-homologs exchange DNA. So here we have the blue one here is longer than the red one. They are not homologs, but for some mistaken reason, they swap some DNA. And I think that ends the chapter. Thank you for sticking with me.